And I'm very excited for today's seminar. And a lot of people know Oliver here, but I'm going to give a little background on Oliver for those who don't. He got his bachelor's degree in 2012 from Newcastle University in Marine Science and Technology. And then from the same university in 2015, he got his master's degree. Then he went on to work at the Cape Eleuthera Institute. And I think you were there, what, 3 years?
Yeah, 2 years on and off.
Yeah, and which was really nice because he then came to SoMAS to get his PhD in my lab group. And that connection with the Cape Eleuthera Institute led to a course that we taught for a few years, and we hope to teach again down in the Cape Eleuthera Institute. And I will say that any of the stories that he tells about me on that trip are not true.
Photographic evidence.
But that was a very nice class that we taught. And then after he graduated here in 2020, he went on to work with Seth Newsom at the University of New Mexico. And he was there for 2 years. And then after that, he went on to be the director of research at Beneath the Waves. And he's been doing that for the last year or so. And along that path, he's been really incredibly productive. Oliver has published 60 peer-reviewed papers for someone that graduated in 2020. That's very impressive, and some very good research. He's also developed a lot of collaborations throughout the world, but also here at Stony Brook. We have people from different departments here that are in the audience here, Greg, Nick, and others. All have collaborations with Oliver, and I do as well. And I should also mention that he is up for consideration for a research assistant professor position here at Stony Brook, and he's given the seminar today. And he's looking forward to building a lab group and research program here at Stony Brook, which I'm very excited about. And I just want to mention before I turn it over to him, along those lines, he was just on a project with myself, Jill Olin, and Bob Serrato, defining foraging hotspots of finfish and sharks in the New York Bight, linking trophic dynamics with spatial temporal trends and species distributions. That was just recommended by New York Sea Grant to be funded. So he's already off to a good start. So with that, I'm really excited to have Oliver here, and I will turn it over to you, Oliver.
Thanks, Mike. Very, very kind words. And it's so exciting to be back. I actually never was able to give a formal dissertation defense because it was in the middle of COVID. So 3 years down the line, it's nice to be able to give a talk back in this room. Can everybody hear me okay at the back? You need to turn down. Yeah, okay. So I'm going to talk about how we can use chemistry. I mean, when I was in high school, I was never really that interested in chemistry, but coming to Stony Brook and working with folks like Nick Fisher and Greg Henkes really kind of revealed the power of chemistry and understanding molecular all the way to kind of ecosystem level processes. So I'm going to talk about a couple of different research projects that we've been conducting over the last, kind of 5 to 10 years, really, and how we can use chemistry to understand kind of cryptic aspects of animal ecophysiology and behavior. So we're going to talk a bit about, we're going to take 3 kind of different leveled approaches, talk about how we can use chemical traces to understand ecophysiological processes that are occurring at the molecular level. We're going to then take a step back and kind of go after how we can use these approaches to understand the ecology of individuals and what that means for kind of diet selection and nutritional condition. And then we're going to take one step back further and look at how we can use these intrinsic traces to understand kind of population and community level processes, particularly as it relates to ecosystem connectivity and resilience. But those of you that know me pretty well know that my tool of choice is that of naturally occurring stable isotope ratios to answer these questions. And a brief primer for those of you that aren't necessarily familiar is in the natural world, we have different kind of forms of elements that vary based on their atomic mass units. So we have hydrogen 1, which has an atomic mass of 1. It has 1 proton, 1 electron. We have deuterium that has a neutron, a proton, and an electron. It's still hydrogen, but it's 1 mass unit heavier. For oxygen, we have oxygen 16, 8 protons, 8 neutrons, 8 electrons. Oxygen 18, 10 neutrons, 8 protons, and 8 electrons. And all we really need to take away from this is that those atomic masses affect how those atoms behave in chemical reactions and in biochemical reactions. What we can do is we can actually measure the ratio of the heavy to light isotopes of the sample, so on organic tissue, relative to some standard, and that gives us value that we typically report in per mil. And the really, really cool thing about these isotope approaches is folks started to kind of measure the isotopic composition of organic material. In food web ecology, we're kind of mostly interested in carbon and nitrogen, and to a lesser extent, sulfur. And when folks are measuring these isotope ratios, we figured out very, very quickly that there are big differences between different kind of taxonomic groups. And so we can tell the difference between C3 versus C4 plants based on the amount of isotopic fractionation that occurs during photosynthesis, right? So in any kind of biochemical reaction, a consumer or a plant in this case is going to preferentially uptake the light over the heavy isotope. And they do that to kind of different extents based on the primary photosynthetic mechanism, and that allows the kind of separation between these different groups. Nitrogen is kind of the same concept. That tells us about nitrogen baselines, so we can infer whether or not a plant is a nitrogen fixer that exhibits very, very low fractionation, therefore ends up somewhere down here, or whether or not a plant is using nitrate metabolism, therefore giving higher amounts of fractionation. And this was great because food web ecologists came along and said, okay, well, what if we start measuring isotope ratios in the tissues of consumers? Well, it just so happens that as you go kind of up a food web, there's very, very little change, or what we call fractionation, in our carbon stable isotope values between a consumer and its prey. And that means that we can basically infer the dominant source of primary production that's supporting a food web. So whether or not a consumer is supported by a C3 plant or a C4 plant, or a mix of both. Nitrogen is slightly different and a little bit more complex because consumers produce a lot of nitrogenous waste, uric acid, urea. And because that product is a product of metabolism, it's going to favor the light over the heavy, therefore resulting in higher fractionation between a prey and a consumer, by about 2 to 5 per mil. This is a really, really basic understanding of fractionation. But for food web ecologists, this was great because we could start to put together these kind of ecosystem level kind of questions on how food webs are structured and how energy was flowing through food webs. And in terms of how isotopes were kind of applied in ecology, as the method kind of started to develop in the late 1970s and early 80s, measurements were challenging, it was costly. But over time, as technology improved, this sort of technique became increasingly accessible. And as a result of that, in kind of the early 2000s, we're seeing an exponential increase in the application of isotopes to answer ecological questions. And that's now mirroring that of kind of genetics that kind of took off in the mid-1970s, which was basically the reason for the formation of GenBank. So we published a paper in 2020 in Ecologia where we were interested in how folks were applying stable isotopes, in this case, to understand ecological niches, so basically the trophic ecology of animals. And we can see here that over time, this kind of red area here is the number of papers using stable isotopes relative to traditional approaches, such as stomach content analysis. So people were really, really starting to adopt this approach. Now, obviously, as things become more accessible, people are trying to push the envelope a little bit. And one of the things that I was really excited to be exposed to during my postdoc was the use of isotopes to measure the isotope ratios of individual compounds, such as, in this case, amino acids. And I'm going to talk a lot about nitrogen today. I'm not going to talk about carbon. But the way that we can group different amino acids based on the amount of fractionation that occurs between a prey and a consumer. And we typically group amino acids based on the amount of fractionation that occurs. So we have trophic amino acids, and these are amino acids like glutamic acid, aspartic acid, isoleucine. They undergo a lot of kind of nitrogen reshuffling during their synthesis. So trans and deamination, the movement of amine groups on and off the molecules, and this results in fractionation. We have another group of amino acids that we call source amino acids that don't undergo minimal trans and deamination. So there's no fractionation associated with that. So all that's to say that in practice, The delta 15N values of our source amino acids are basically telling us what's going on at the base of the food web, that nitrogen baseline that I was talking about in my previous slide. Whereas these trophic amino acids are going to scale with an animal's trophic position, basically. And so what people started to do was that they started to look at the differences in isotope values between these trophic and source amino acids, the differences between glutamic acid and phenylalanine in this case. And what this meant is that you didn't actually have to go out anymore and measure the nitrogen isotope values of plants and food webs. You could just look at your phenylalanine value of the consumer and match that to its glutamic acid value to come up with an estimate of trophic position. As you can see here, there was a couple of papers that have done that, looked at trophic position estimates of tuna, tiger sharks, sculpin, moray eels. But the one thing that food web ecologists often forget is that we're dealing with a system that is inherently dependent upon physiology. And I actually think in terms of amino acid isotope values, yes, you can go out there and you can measure something's trophic position. But there are really interesting and nuanced physiological questions that you can answer with this technique, far more so than ecological ones. And there was a really cool paper that came out by one of my colleagues in 2021 by John Whiteman, where he was using amino acid isotope values, nitrogen isotope values, to understand the energetic status of different animals. Can we detect things like tissue catabolism, so periods where animals will actually break down their own tissue stores to create new tissues and build new proteins? And so what he did is he measured the nitrogen isotope values of a capital breeder, in this case, emperor penguins. And he was looking at the differences between those trophic and source amino acids between different tissues. So he was looking at muscle tissue in this case, which is a really slow, it has a very, very slow rate of turnover. So that means it's reflecting kind of dietary information across a really broad range of time. He was comparing that to reproductive tissues. And what he found was that there are big isotopic differences between proline and phenylalanine in muscle relative to those reproductive tissues. So the isotope values of reproductive tissues were a lot higher than that of muscle. And typically, if we're thinking about this in a food web context, we would interpret that as a difference in an animal's trophic position, because glutamic acid and proline are responding to differences in of an animal's trophic level. But what's going on here is actually emperor penguins, which are capital breeders, so they feed and then they stop feeding during their kind of reproductively active periods. They're actually utilizing and breaking down their own muscle to build reproductive tissues. You then compare that to an income breeder like a herring gull that feeds constantly, regardless of what reproductive state it's at, and we don't see any differences between the muscle tissue and those reproductively active tissues. So what he's done here is he's basically figured out, using the nitrogen isotope values of individual amino acids, a proxy for capital versus income breeding. And this idea of kind of breaking down your own tissues to make new ones and being able to detect that using nitrogen isotopes was also pretty apparent. It's a relatively universal thing amongst vertebrates, because many different vertebrate taxa undergo these periods of kind of fasting versus active foraging. And so we can see, in this case, these were elephant seals from Antarctica. One of my colleagues, Nicky Lubka, published this in 2020. We can see, actually, the zoom window at the top there is hiding the. There we go. So you can see there that during periods of active fasting, the nitrogen isotope values of all of these individual amino acids are often higher than that of active foraging, illustrating that these animals are liberating amino acids from structural tissues and building them into new ones during these periods of nutritional stress. So all of this kind of put a spoke in the work of traditional food web ecology, because there's such a huge physiological imprint on isotopic variability, at least with regarding delta 15N values of individual amino acids. So I went into my postdoc and I was talking to Seth, and we were like, okay, well, let's investigate this nitrogen balance idea. Well, let's pick a group of animals that are inherent, probably one of the most nitrogen limited groups of animals on the planet. Just so happens that I've done quite a lot of work on sharks, and we had some good access to samples. So we were interested in whether or not we could pick up this, kind of physiological imprint on an animal that is, one, completely nitrogen limited, and two, during a period that is of, resulting in incredible nitrogen demand, such as pregnancy. So for those of you that aren't familiar with shark physiology, they're a great physiological model for a bunch of different reasons. But they have extremely high demands for nitrogen because they utilize urea to maintain osmotic balance, so to reduce water loss. So they have an additional demand for urea. Urea is full of nitrogen. They also have an additional demand for nitrogen because there's these really interesting relationships between nitrogenous compounds such as trimethylamine N oxide and urea, which marry together in order to stop negative impacts of hydrostatic pressure on cell homeostasis. Sharks are actually found all the way from coastal oceans down to about 2,500 meters. And the ratio of TMAO, in this case urea, is really important to help them live in really deep water environments. So they need a lot of nitrogen. I'm not sure if any of you are familiar with the story of the bonnethead shark, but this was a species of shark that is interesting for many different reasons. But they're actually found to consume large amounts of seagrass. So they're one of the first omnivorous species of sharks. So they're actually able to assimilate nutrients and protein directly from consuming seagrass, which was pretty unheard of across a lot of different vertebrate taxa. But from a reproductive and a nitrogen balance perspective, they're interesting because they undergo what we call this exhibit hybrid vivipary. And what that means is that as they go through pregnancy, their embryos are sustained by a yolk sac placenta initially, and then when they get to about 9 to 10 centimeters, that yolk sac turns into they're sustained by a yolk sac until they get to about 9 to 10 centimeters. That yolk sac then is converted into a placenta when they get to that size. So you have this period of kind of an inactive relationship with mum in terms of its nitrogen balance, and then once that placenta is formed, you have direct nutrient transfer between the embryo and the mum. And then when they get to about 25 centimeters, they give live birth. So the question that we were interested in here is, typically when we're thinking of nitrogen isotope values of individual amino acids, if I wanted to go and calculate something like a trophic position estimate from a pregnant shark, how much of that value is affected by basically the pregnancy stage of the animal? Are animals basically shifting their nitrogen balance during different periods of pregnancy? Can we pick up that signal using our amino acid isotope values? And do we see differences between tissues that kind of turn over faster or turn over slower? So what we're effectively trying to do here is develop a pregnancy test for sharks using this approach. We're fortunate that and this is one of the great things about, if you're ever out there collecting field samples and you don't know what you're going to do with them at the time, please keep them, because people will eventually come along and say, oh, do you have any tissue samples from this species of fish over the last 20 years? And they'll be able to use them for really, really interesting questions. And so we were able to take advantage of a sample set that was collected almost 25 years ago by a NOAA gillnet survey that went around and were doing gillnet surveys for bonnethead sharks in a bunch of different estuaries west of the Florida Panhandle. And we sampled individuals across a broad range of estuarine environments to try and account for differences in the nitrogen baseline that might occur. So you could have different nutrient dynamics in all of these different areas here, which can affect your delta 15M values, basically. And one of the overwhelming things about using amino acid approaches, isotopically, is that instead of just getting one value, you actually end up getting about 12 values, because you've got a delta 15M value for 12 different amino acids, times by 25 individuals, times by 2 different tissue types. So it's a lot of data that comes off the machine. And so one of the things that people typically do is they use these kind of dimension reduction techniques, such as principal component analysis, linear discriminant analysis, to distill that information down into two different axes here. And what we're interested in is whether or not the isotope values of these trophic and source amino acids are varying between early term and late term individuals. Early term individuals are those mums that have embryos inside of them that are reliant upon the egg yolk, and the late term individuals are the mums where that placental connection has been formed and is established. What we can start to see, at least for the liver tissue data here, we can start to see that there's separation between early term individuals, telling us that there's differences in isotope values between those two stages. And this, I promise there's not going to be like heavy statistics in this talk. But what we did was we ran some generalized linear models. And what we're interested in here is basically what is driving those isotope values in those pregnant bonnethead sharks. Is it the fork length of mum? Well, we typically see ontogenetic diet shifts in animals with size that can have an effect on 15N values. Is it the average fork length of the embryos? So is it basically the gestational stage that the animal is at? Is it the number of embryos inside mum, or is it some kind of interaction between body mass and the length of mum as well? And what happened is, what we started to see is we started to see this negative effect of the fork length, of the embryo fork length. So what this meant is that those isotope values, those 15N values, are going down as animals proceed through pregnancy. And again, those trophic source pairs that folks often use to understand things about the length of food webs, well, we're actually seeing them differ between early and late term individuals. Those isotope values, as an individual proceeds through pregnancy, are starting to become a lot lower. And we would, originally, if we didn't know anything about the reproductive biology of these animals, we'd interpret that as a shift in its diet or a shift in its trophic position. But what's happening here is we think that these animals are actually shifting their nitrogen balance. So the ultimate sources of nitrogen that are occurring during pregnancy. And the interesting thing about this is that relationship is completely opposite to what you'd expect during, for a catabolic state. If an animal is breaking down its own tissue, those isotope values usually go up, and we're seeing them go down. And so we came up, this is one of the reasons why this paper was a little bit challenging to get through review, because the reviewers were like, well, this is really cool data, but you can't actually test any of these hypotheses that you think might be going on with bonnet head sharks. So we came up with 3 ideas that could potentially explain why those isotope values are going down as animals kind of proceed through pregnancy. The first one, and probably the most likely, is nitrogen retention, so reducing the amount of nitrogenous waste that you're making. But these last 2 are, in my opinion, the cooler ones. And one of them is nitrogen scavenging. So there's theories to suggest that sharks can actually absorb nitrogen over their gills. And the last one is the bacterial breakdown and liberation of nitrogen from urea. I can see Nick shaking his head at some of these, so maybe he agrees. He doesn't agree with some of them. But this is probably the most likely one. And we see as animals proceed, humans proceed through pregnancy, they reduce the amount of nitrogenous waste. So you reduce the amount of urea. Because urea is isotopically light, that gives isotopically light nitrogen back to the pool from which you can then synthesize your protein. And we see the same if you look across a longitudinal record of a pregnant human's hair as they proceed through pregnancy, we start to see the delta 15N values going down. And that's because they're reducing their nitrogen excretion. Hopefully I can convince you of this, Nick, because this is quite an interesting story. There were some really cool papers that came out in kind of the mid to late 2000s on the ability of elasmobranchs in particular to scavenge nitrogenous compounds from the ambient water. And in this case, dogfish sharks were basically assimilating ammonia over their gills and using that nitrogen to route directly into protein synthesis. So basically eating over your gills. The question is whether or not this is enough to maintain the nitrogen demand of an animal during pregnancy. What we can see is the ammonia concentration of the ambient water column goes up, the ammonium uptake rate and the amount of nitrogen that's liberated from that ammonia and built into tissues also increases as well. The last one is whether or not there is microbial breakdown of urea, which liberates this isotopically light nitrogen that can be rerouted back into protein synthesis. So a lot of shark tissues have a ton of bacteria all over them. They have this enzyme called the urease enzyme, which basically can break down urea molecules and liberate nitrogen, which can find its way back into the nitrogen pool to then go and synthesize your amino acids and proteins from. So there's 3 pretty interesting theories that we think, it could be a combination of them, or it could just be, one. But the main thing to take away from this is that these amino acid isotopes, historically we wanted to apply them in a food web context because that's what ecologists like. But they're actually, in my opinion, far more of an approach to really understand patterns of animal ecophysiology. How are molecules and how's the demand for molecules shifting during, periods of starvation or pregnancy? We can actually start to trace that. And we also were able to run deuterium isotope values of individual amino acids. And I'm going to put myself out on a limb here because my knowledge of hydrogen physiology is nowhere near that of nitrogen. However, hydrogen is telling us basically that there are different sources of hydrogen, whether it's from your food, whether it's from your kind of metabolic water that's formed. And we can see that animals that are fed kind of different diets can often have different amino acid values of deuterium, which is reflective of how much hydrogen they're deriving from water versus, carbohydrates, proteins, and lipids. And what we also saw, and this was kind of crazy because those of you that have ever measured hydrogen isotope values before, it's a really difficult measurement to make, because your machines are sensitive to things like humidity and how much moisture is in the air. The values can be super variable when they come off the mass spec. But we see these pretty consistent declines in our deuterium isotope values of our amino acids as animals, again, proceed through pregnancy. And when we plot the isotope values of proline versus glutamic acid, pretty hard to see on the board here, but we see absolute separation between early term and late term individuals. So the cool thing about these kind of molecular isotope-based approaches is we can start using them to really infer, really important processes occurring at the molecular level, rather than just what trophic position an animal is sitting at, which I don't think is anywhere near as interesting as to what's going on with hydrogen or nitrogen biochemistry during physiologically demanding events. So that's all the kind of heavy molecular stuff. We're going to take a step back now and talk about a project that is a little bit more closer to home. It involves sand tiger sharks in New York estuaries, and how we can use, in this case, bulk isotopes to understand linkages between individuals' preference for diet and diet selectivity, and what that means for their kind of energetic condition. So again, a kind of a physiological spin on an ecological question. And sorry if this is a bit cliche, and I really don't like talking about doom and gloom, even though it's an important thing to consider, but things are changing, more hurricanes, waters are warming, we're getting more fires, glaciers are melting, it's all really bad. And one of the main things that we're concerned about, I suppose, along the coast of the east coast of the US is how that's impacting the distribution of prey and predators. And Mike was mentioning we just got a proposal funded by New York Sea Grant to kind of understand how animals are selecting foraging locations and how that's likely to change as prey distributions change well into the future. But one of the things that we've been kind of historically bad at is understanding what that means for actual fitness and longevity. How is the nutritional or the energetic condition of a consumer affected by having to switch its diet necessarily? And there was, I'm sure some of you have probably heard in the early 2000s, there was this huge die-off of stellar sea lions that occurred in the Aleutian Islands and near Alaska. And at the time, people were wondering, well, what's causing all of these die-offs? And over the last 20 years, we developed a pretty good understanding is that these individuals were forced to shift their diets from lipid-rich prey, such as herring and sandlands, to what we call trash fish, things like cod and pollock that aren't necessarily energy dense. And so it's obvious that there are potential connections between an animal's diet and what that means for its longevity, fitness, and other kind of long-term impacts. And we had a really unique opportunity to study juvenile sand tiger sharks here on Long Island. And we were interested in, because when animals move into these estuaries, they have a broad array of prey items to choose from. So we were interested in, okay, well, how broad is their diet? But more interestingly, how does that impact the concentrations of recirculating lipids in their blood plasma, things like triglycerides and cholesterol? How variable are those values? And are they linked to the dietary decisions that animals make? And so for those of you that aren't necessarily familiar with their migratory history, when the water's warm, kind of at the end of May, beginning of June, these sand tiger sharks move up into these Great South Bay estuaries and are sustained by estuarine productivity for the majority of the summer. They then move out of the estuary back down to the coastal Carolinas. We've worked with the Wildlife Conservation Society for a really long time, who have been historically sampling these juvenile sand tiger sharks for the last kind of, I suppose, 10, 12 years now, Mike, something like that. And they had an absolute bulk load of blood plasma. And so we were interested in a couple of questions. So we're interested in obviously how the diets of sand tiger sharks impacted their nutritional condition. But for us to get there, one of the things that whenever you're measuring isotope values of a highly migratory consumer, you have to make sure that the isotope values that you're measuring in the system you're measuring them in are reflective of those local conditions and not something that happened, before the animal moved or migrated there. Right, and so this is from one of my PhD chapters that we published in Methods in Ecology, but we derived something called an isotopic clock to figure out how long each sand tiger shark had been inside that estuary before we'd sampled it. So we could be sure that individuals were at what we call isotopic steady state with that estuarine system and weren't reflecting foraging that happened before they arrived. So this model is relatively simplistic and it only requires 4 different parameters. And what we need to know is we need to know what a sand tiger shark should look like isotopically at steady state with the estuary. So we had some prey values and we were able to kind of predict that. We need to know, and so that's kind of this delta F parameter here that you can see in this model. This is just a basic rate equation. We have these delta 0 parameters that reflect the what a sand tiger shark looks like isotopically when it's off the coast of the Carolinas and it's feeding in that system. We have delta T, which is the isotope value of the sand tiger shark when we measure it. And we have this lambda parameter here, which is basically, we call the rate of isotopic incorporation. So if I'm feeding in one area and then I move over here and I start feeding again, how quickly are my tissues going to reflect that change? Feeding here relative to there. And we know for blood plasma in sharks, it's about 40 to 60 days. So what we do is we can plug all those parameters into a model and come up with a residency estimate for each individual in the Great South Bay. So what we can do is we can figure out how long it's been since each of those animals have migrated from outside the estuary inside the estuary. And we can remove those individuals that we don't think are at steady state with that estuarine system. The next thing we do, okay, so we know our sand tiger shark, the sand tiger sharks that we're sampling, they're reflecting these local estuarine prey values. The next thing we need to do is actually figure out what the prey look like isotopically. So we picked 3 different prey groups in this case. We picked pelagic forage, so that's things like menhaden and scup. And their isotope values are pretty distinct because they reflect C3 phytoplanktonic primary productivity, so they give very, very light or negative isotope values, at least in carbon space. And that's slightly different from things like benthic detritivores and demersal omnivores. So what we're doing here is we're basically building what we call a prey mixing space. And then we're going to use a model which figures out the relative contribution of this, this, and this to each individual. And what we saw here, because individuals were plotting relatively close to these 2 prey groups, so that's pelagic forage down here and demersal omnivores, we actually removed the benthic detritivore group because we didn't think it was very important. So our isotope system is set up. The next thing we had to do is measure the recirculating concentrations of these different plasma lipids. And in elasmobranchs, we think that high levels of triglycerides, cholesterols, and these kind of molecules that move these lipids around, like low-density lipoproteins, very low-density lipoproteins, we think higher concentrations are reflective of individuals at better nutritional condition. There are some cases in vertebrates, things like when bears go into hibernation, you'll see their triglycerides skyrocket because they're breaking down their own fat. Sharks don't do that. So we think that higher triglyceride concentrations in this case are more reflective of healthier individuals. So what we can do is we can build all of that information into what we call an isotopic mixing model. And all that's doing is it's telling us the relative contribution of each of these prey groups to an individual's diet. And we can now add covariates into this modeling framework. So we can see how the contribution of different diet groups actually starts to impact the concentrations of these recirculating plasma lipids. And what we found was that individuals that are consuming, these are two different size groups of animals, as we know that size can also impact isotope values, so we tried to remove that bias. And what we found was that individuals consuming larger amounts of pelagic forage fish, so those are the more kind of energy dense fish, have higher levels of cholesterol, very low density lipoproteins, and triglycerides. And this is some of the first evidence to really kind of use isotopes and other physiological markers in a way to understand the energetic consequences of diet variation. The one thing that we weren't able to do, and this is a real shame, is that all of these individuals also have internal tags that can be used to track their movements up and down the coast. And we weren't able to use the data in this case to figure out whether or not all of this actually starts to impact how animals are moving. So what we're finding here, we're finding that we can use isotopes and we can use other kind of nutritional markers to understand how diet selectivity might impact energetic status. But we're not able to necessarily piece together the impacts on fitness and movement. And that's something that I'd really like to start building on to some of the sand tiger work that we've already started off New York. However, we do have some evidence from fish. This is the only freshwater example I'll talk about today, but we have an example from lake trout from a lake of Two Rivers in Ontario, Canada, which is a beautiful, beautiful lake. And we're finding that, so this is another kind of dietary proportion estimate. We were using amino acids here, but what we're interested in is whether or not the contribution of aquatic primary productivity, bacteria, and a lot of C3 productivity, as those production sources change, does it impact fitness? And obviously, I would probably ignore the right-hand side of these figures because obviously, as your sample size diminishes, your error inflates. But what we can see in the first half of these figures here is that as our contribution of these different energy channels changes, it starts to impact the relative mass of the fish gonads. So what we're starting to see is that there are potentially not only energetic consequences, but also consequences for longer-term fitness based on how energy is kind of transported and assimilated through food webs. Jen, this is something that we were talking about earlier on. I think it'd be really cool to apply to the Hudson River data set. And finally, we did some work on permit off the coast of Florida, and here we were interested in whether or not the kind of movement component. So what we've got here is we've got estimates of whether or not individuals were deriving the majority of their diet from seagrass-derived ecosystems, also seagrass-supported ecosystems, or pelagic ecosystems, which you can see in the blue, and whether or not that was predicted based on movement. So these data points here are basically average movement trajectories for 109 different fish. So where they're spending the majority of their time, basically. And you can see, and what this kind of came out to be a really nice spatial interpolation, because we can start to see that where you find animals along the coast actually starts to imprint on the amount of energy that they're deriving from ecosystems that are available to them. So the main takeaway from this is that there is also implications for animal movement in addition to nutrition that comes off diet selectivity. So we've got a project down in the Turks and Caicos Islands where we're trying to pack all of this stuff together. We're trying to figure out how animals are using energy from different systems, how that impacts movement, and how that impacts kind of long-term fitness and longevity. Okay, how are we doing for time? Okay, we're good. So we've gone from the molecular level to the individual level. The last thing I want to talk about today is how we can use these traces to look at kind of more 30,000-foot view community and population level approaches, particularly how it relates to the functional role of animals and how animals might move nutrients between different ecosystems. And the motivation behind this is the fact that animals move, right? Animals move incredible distances, whether or not it's a tiger shark moving up and down the coast of the US, whether or not it's a humpback whale moving between the subtropics and polar ecosystems to facilitate different parts of the life history. Animals move, and these movements have occurred for millennia, and they're really, really important in terms of nutrient transport. However, in today's world, when we think about animal movements, we think about all the negative stuff. We think about, okay, well, this animal moves fast, so what's its likelihood of being impacted or caught by in a commercial fishery? And how is that impacting population dynamics? And while that's important, so for example, this was a paper that was published in 2019, which was looking at the overlap of commercial fisheries with, I think it was something crazy like 1,200 satellite tag sharks. And they were looking at the likelihood of fisheries capture. But these long distance movements, while they might have negative consequences for animals, are actually really important for kind of oceanographic processes, because large animals, especially things like whales, they feed in one area, they poop in another. And that moves nutrients between both vertical levels of the water column and also horizontal levels of the water column as well. So in terms of, and these are important for moving things like iron. This was a really cool paper that came out in 2016, which described the role of whales in transporting 340 million kilograms of phosphorus per year between different ecosystems, and everything's connected. What goes on in the ocean, as we know, can affect things in freshwater and aquatic ecosystems, which can affect things on land. And one of the things that we don't really understand, particularly, is the role of these mobile animals in actually connecting different ecosystems. Nobody's actually gone out there and really done a thorough job of measuring it at any sort of great scale. And there was a really cool paper that came out in 2012 by Douglas Macaulay in Ecological Applications, where he was at Palmyra Atoll, which is this remote atoll in the middle of the Pacific Ocean. And there are three major habitat types. There's lagoon ecosystems, there's fore reef, and there's pelagic ecosystems. And you have three predators here, red snapper, blacktip sharks, and gray reef sharks that move consistently between all of those ecosystems. And so the question was, what proportion of energy was a highly mobile animal deriving from each of these available systems? And he did exactly the same thing that we did with the sand tiger sharks, and found that each of these individuals were utilizing prey that was deriving energy from at least two of these different systems. And that was used as a proxy for ecosystem connectivity. So one of the big takeaways from that was that these highly mobile predators are actually pretty important in moving nutrients in horizontal space. And that has huge implications for things like resilience, because there's some really cool theoretical work out there that basically states that animals that kind of connect different food webs in space and time are really important for food web stability. And this is a really underrepresented and a really understudied component of food web ecology. And for the last 10, 15 years, I've been really fortunate enough to work in areas such as the Bahamas. And while the Bahamas are stunning for a bunch of different reasons, they're really cool ecologically, because you can go from a mangrove creek to 800 meters of water in less than a mile of horizontal distance. So you have all of these ecosystems that are smack bang next to each other. You've got mangroves, seagrasses, coral reefs, open ocean. And there has to be huge energy flow that's occurring between all of them, if not from the tides, from the movement of highly mobile animals. And we know from things like satellite telemetry that these animals are not only moving horizontal distances, but they're also moving in vertical distances as well. So this is a Caribbean reef shark. Caribbean reef sharks typically are found on coral reefs. But some satellite tracking data that we published in 2017 shows that these animals actually move down to 200, 300 meters of water. So they're moving off the shelf and they're migrating up and down. So and this was a paper, here's Mike, back in 2015, but this was a paper that I wanted to write for 10 years. We just didn't have the data to be able to do it effectively, but now we do. And we've been sampling tissue samples from sharks in the Bahamas for the last 10, 15 years to kind of get at this ecosystem connectivity question. And this was when Mike first came down to Eleuthera back in 2016, I think. And these samples have been collected as part of Stony Brook field courses as well. So it's kind of really nice to come home and talk about this, because there were a lot of students that were kind of critical to the sample collection that was required to work on this study. So this was when we taught MIR 532 back in 2019. So the first field source we taught. And so the main question here was, how do highly mobile animals utilize the major energetic resource pools that are available to them? And if they're using multiple, how is that impacting ecosystem connectivity? So what we did is we built this, again, another what we call isotopic mixing space. This paper was just actually published in ProcB, and I apologize because I don't know if any of you have published in ProcB recently, but they really do try to ruin your figures when it goes to the proof stage. So we developed this isotopic mixing space. We were interested in how 17 different species of elasmobranch were utilizing seagrass, oceanic forage species, coral reefs, and large oceanics. And what we did is we developed we built basically prayscapes for what the prey of the sharks would look like if they were feeding on a coral reef, if they were feeding in seagrass, etc. And we then use that Bayesian mixing model approach to understand the relative contribution of each of these sources to biomass of the animals. Right? And so we had 17 different species occurring in neritic ecosystems, some of them that were oceanic, and all the way down to deep sea animals. So we had a really kind of unique data set of species that resided on the coast all the way down to 1,000 meters of water. And what we found was that, unsurprisingly, all of these animals were reliant upon at least two of these different resource pools that were available to them. Okay, so all of these 17 species of elasmobranch were really important for connecting different areas of the seascape, all the way from the coast down to the deep ocean. And what we did is we took this one step further, and we took all of those contribution estimates to those different 70 different species, and we plugged it into a cluster analysis. And what that allowed us to do was to pick up different functional groups of species based on the degree to which they were connecting different regions of the seascape. So we had what we call neritic ecosystem connectors, and these were species such as nurse sharks and stingrays that were important for connecting seagrass and coral reef habitats. We had neritic to oceanic connectors, things like lemon sharks and Caribbean reef sharks that were important for connecting neritic habitats with the open ocean. And then we had offshore animals that were kind of more responsible for connecting different components of those offshore food webs. And then we were also able to derive a higher oceanic predator group that were exclusively reliant on offshore kind of large fishes. And we also ran this thing called an evenness analysis, which basically was able to tell us the degree to which animals were connecting different ecosystems. So basically, it's a scale between 0 and 1. Animals that are down here at 0 are reliant upon only a single ecosystem. Animals that are up here at 1 are reliant upon multiple. And we can see every single species in this analysis was reliant upon more than one ecosystem, therefore providing inherent connectivity between them. And we're really excited about this, because it's one of the, like I said, it's a paper that I wanted to write for 10, 15 years. And we're able to, for one of the first times, to take these estimates of kind of proportional use of different resource pools and plug it into an analysis to derive an estimate of an animal's functional role, and actually group species together based on their functionality. Typically, when we think of functional roles, we think of things like trophic position and things like that. But we're now able to group species together based on the degree to which they connect regions of the seascape, which is quite exciting. And we also found that this, at least in subtropical locations, elasmobranchs are really important for connecting neritic ecosystems all the way to the deep sea as well. So this has obviously huge implications for resilience. As these low-lying, developing nations are going to be at the forefront of climate change and habitat destruction, sadly, over the next 50 years. And this idea of habitat connectivity is something that we're also looking at throughout vertical areas of the water column. And part of a study with one of my colleagues, Dan Madigan, who I'm sure some of you know, who worked at Stony Brook. And we're interested in this relatively new isotopic tracer that uses isotope ratios of mercury. And again, I'm not a mercury biogeochemist, so bear with me. However, what we see in surface lit waters with a lot of light, it causes what we call photodemethylation, and that has an isotopic effect. So animals that are deriving their energy from surface waters have really high mercury isotope values. Animals that are deriving their energy from mesopelagic waters, where there's a lot less photodemethylation, have low mercury isotope values. That's the way so with that, hopefully 50 minutes, not too bad. I'd like to thank all my collaborators at UNM, NOAA, and Beneath the Waves, the Cape Eleuthera Institute, and obviously folks here at Stony Brook. And with that, I'll take any questions.
Thank you, Oliver. Do we have questions?
I tried to pack a lot into that, so sorry if it was a lot of information all at once. Yeah.
You covered a lot of ground. Is that an understatement?
Yes, I tried to.
Yeah. A lot of questions. I wanted to raise something which is you didn't discuss at all, and that is that one of the largest patterns we see, which is claimed to be, and that is exchanges between estuaries and the open sea, and
right.
Especially there are latitudinal differences, like anadromous versus antagonist fish.
Yeah.
So have you ever done any work to try to look at that? Because there have been claims about the trophic values of the open sea versus estuaries with latitude that, as far as I know, have never been tested very well. but they're claimed to be true, and yet this seems like a possible way to address it.
Absolutely, I think that estuarine systems are just one of the most complex ecosystems to study in general, because you've got, sometimes fluvial runoff that's impacting them. You have stuff, coming in from the shelf. And there's that isotopic clock model that I jumped over pretty quickly there. They originally used that to look at estuarine residency in migrating salmon. But so is the question, how does the trophic dynamics of species between the shelf and estuaries change latitudinally?
Yeah, that's one of many problems you can solve.
I mean, you could absolutely resolve it. I think you could resolve it more so with the amino acid approach, probably, than you could with bulk isotope ratios. You'd also have to start using things like sulfur. And sulfur is really good for looking at pelagic versus benthic productivity. So absolutely, I mean, that's definitely something that we should start looking at. Honestly, we have the ability to do it, so we should.
Jeff, I actually had something very similar, and we'll talk about this later, but some of those anadromous fish that you're talking about that have go from interpairs to semipares up and down the East Coast, and semipares fish are actually consuming their body to the extent that when they spawn, their organs are trashed because they consume them. To spend all their energy to spawn once and die, because the interpairs fish are spawning multiple times. So I was actually thinking about similar along these lines, but when you're talking about catabolism and ecophysiology and whether we can see markers for that. But we'll talk about that later.
A really interesting area to dig into, Jeff, is are you familiar with Kevin McCann's work on like diversity, stability and stuff like that, and looking at the role of slow versus fast energy channels and contributing to ecosystem stability? Because what you can do, especially with the amino acids, is look at the contribution of kind of the microbial loop versus other, faster turning over energy channels, and how that might impact connectivity and stability of estuarine systems, and how that might vary with latitude. So the world's our oyster and wide open with those sort of questions. So definitely something to consider.
Jan?
Yeah, thank you. Great talk.
Thank you.
And I see a lot of potential for, applying some of the approaches to Hudson River samples that we collected. So my question to you is that because a lot of us with isotope reflect the long-term accumulation, right? And it's not like where you sample them.
So
It's a cumulative impact.
it depends on which tissue you're measuring the isotope ratios in, because if you have, some of the work that me and Greg were working on was, for tissues that turn over really fastly, but they're quickly, that have a fast metabolic rate, things like tooth collagen. Those are giving you what's going on within the last 30 days versus something like muscle, which is kind of a time integrated average over the last, at least in a slow growing fish, like 6 to 8 months.
Those are varied among species.
Varies among species, varies among sizes, and it varies among tissues, among also things like the water temperature that the fish is living in, all of that sort of stuff drives.
Yeah, so my next question is about life history stages. When you look at all your samples, did you divide it into the different life history stages, look at the differences?
For the sand tiger sharks we did, yes, but they were all juvenile and subadult, so they're all kind of the same life history stage. For the sharks in the Bahamas, we were using muscle tissue, so that's really slow turnover rate tissue. We were trying to get rid of any seasonal differences that you might see. But the subtropical and tropical food webs are, yes, the seasonality, but it's nowhere near as strong as somewhere like the temperate Northeast Atlantic, and so it's more of a consideration here than it would be, down in the Bahamas, for example. So,
Yeah, thank you.
We have a student question.
Hi, great talk, super interesting. So I'm interested in that isotopic clock thing that you mentioned. You did mention to be pretty interesting for whales as well. So I guess sort of methodological questions. One, what type of tissue is absolutely necessary? And two, how precise regionally do you think you could constrain it? We have, humpback whales off here on the South Shore that we've been sampling for a couple of years now, and we have no idea how long they stay here. Are they going up to the Gulf of Maine? Do you stay here year round? When do they migrate? Stuff like that. So I was curious.
yes, so you could use any tissue. So what happens is when an animal goes through a diet shift, the majority of the change in the isotope values will occur in a relatively quick period of time. So we call that, we've got T50. It's time to half turnover in this case. So even for muscle tissue, even though it turns over slowly, you can still detect the signal. And me and Brandon actually have been working on and have developed an R package called IsoClock R, which is on GitHub, I think, right now. But we still just, it's one of those papers where it's like we need just like 2 days to sit down and finish it off. But it provides all the bells and whistles that you need to be able to construct an isotopic clock for a whale. He's also included some really, really cool, like, I'm not a statistical whiz, evidently, Brandon is. And he's included all these really cool bells and whistles, things like rejection sampling, to really make it really statistically robust in terms of the estimates that you make. So we should definitely apply it to whales. I can, yeah, if you want some advice on that, I can definitely. So long as there's a gradient between where the animals are coming from and where they're staying, you can run the isotopic clock. Great. Thanks.
Okay, so we have time for a couple more questions. Bob, I've been told that I have to go with students, so granted. They're coming to get Bob. You're not Bob. The Brandon and Bob.
Great talk, Ollie. So a lot of you have to have a lot of knowledge about the kind of isotopic composition of prey and mesopelagic species, bristle mounts, for instance, super abundant.
Yeah.
Within an individual, you can have huge oxygenic variability that can also be latitudinal, whether they're super lipid rich or they have a gas-filled swim bladder. I guess how much information is kind of known, because I haven't found many studies that have really actually hammered out like isotopic measurements from big tovids, bristle mouths, kind of.
Yeah, so the good thing, well, I mean, mercury is going to reflect, and maybe Nick is probably more better to answer this question than me, but there is definitely latitudinal variation in mercury isotope ratios that are reflecting local conditions. So if you're thinking about the contribution of mesopelagic versus epipelagic, then there is absolutely regional variability as you go down latitudes. Now, if you're talking about how much variability there is in carbon and nitrogen isotopes of McTophids, we have no idea. And deep water ecosystems are of extreme interest, to me, and that mesopelagic component is really poorly studied isotopically, and it's just an artifact of the amount of sampling that goes on. So if you're bringing bristle mouths up in the offshore survey, then we should absolutely start measuring.
Well, show us that we have a few thousand like big tovids.
So there we
bristle
go.
mounts.
Yeah. So yeah, absolutely.
Bob?
So that was a fantastic talk, covering so many topics. There are a million questions we could ask, but I'm going to ask kind of a mundane question, which is, you refer to ecosystems and connectivity between ecosystems, but when does the connectivity become so great that it is the ecosystem, that ecosystems meld into a single system? And how do you decide what's a part, what?
So I think the next, we're kind of scratching the surface here with the isotopes. We're basically saying, okay, well, yeah, there's connectivity there, and these are the species that are doing it. What I really want to do is start putting a value on the amount of carbon that's exchanged between one system and another through predators. And until we know that question, we can't necessarily say whether or not seagrasses and coral reefs become one. But when you scuba dive in these areas, as soon as you get off the reef, you're in seagrass. So, that's why I kind of describe it in terms of seascape connectivity, because the reality is, even though you've got different sources of primary productivity, everything is basically one big ecosystem, including the deep ocean as well. So, and their work in the Bahamas is interesting because, they're really looking to start to develop offshore marine protected areas as well. And one of the things that you have to do to establish that is prove this connectivity with areas that are already protected. And in this case, there is. So, yeah.
Okay, so we'll take one more time, Bob.
So you're talking about fairly large scale systems. And has there been work done at a smaller scale level, like within an estuary, to look at, say, bottom feeders that are sampling in different types of habitats? And is there a way to perhaps get at how much time is being spent foraging in different patches?
Yes, there is definitely work on that. The issue is whether or not those microhabitats differ enough, at least the stuff that I've been talking about isotopically. And typically what's driving, the fundamental isotopic variability is, photosynthesis, it's, the role of bacteria in some cases if we're looking at sulfur. But sulfur is an interesting one, and that could definitely tease out differences in habitat type, especially if your availability of oxygen is different in those systems as well. If you've got sulfide versus sulfate metabolism. So again, we should measure it, let's figure out.
Okay, well, thank you, Oliver.
Thank you.